Hello friends, welcome to my channel. This is MEDICOLINK and I am Dr. Prashant Gupta, and today we are going to discuss on iron deficiency anemia. So let's start. Iron deficiency anemia is the most common nutritional deficiency anemia in the world and in India. So what are the causes of iron deficiency anemia? So iron deficiency anemia can be due to decreased supply of iron or increased demand of iron, or it can be due to blood loss, or it can be due to intravascular hemolysis. So when there is an imbalance between demand and supply, it will result in iron deficiency. Low supply of iron can be seen due to nutritional deficiency, which includes prematurity, poverty, and iron deficient diet. Iron deficiency can also be seen in malabsorption like celiac disease and post-gastric surgery. Increased demand of iron can also be seen in pregnancy, lactation, and iron is also required for growth. So in these cases, there is increased utilization of iron. Iron can also be lost in case of blood loss, as in case of gastrointestinal bleeding like peptic ulcer disease, Meckel's diverticulum, hookworm infestation, or blood loss seen in case of colon polyps or cancer of colon. Loss of iron can also be seen in case of intravascular hemolysis, where there is destruction of RBC. So we can see intravascular hemolysis in case of paroxysmal nocturnal hemoglobinuria, microangiopathic hemolytic anemia, etc. Most common presentation of iron deficiency anemia is fatigue and irritability. It can also be presented as latent iron deficiency anemia. This is a lab diagnosis where there is deficiency in iron without causing anemia. So there is iron deficiency without anemia, which means there is low iron store. So there is decreased ferritin level, but the hemoglobin level is normal. Iron is required in pediatric age group for brain development up to 4 years of age. So in this patient, you will see pica, that is increased tendency of child to eat non-edible stuff like soil, clay, etc. So this is pica. It can also present with Plummer-Vinson syndrome. Plummer-Vinson syndrome is characterized by a triad of microcytic hypochromic anemia, atrophic glossitis, and esophageal wave. So this forms a triad of Plummer-Vinson syndrome, that is microcytic hypochromic anemia, atrophic glossitis, and esophageal wave. Now we'll talk about iron metabolism. Now talking about iron metabolism, iron is absorbed in the form of ferrous iron, that is Fe2+. Iron is absorbed in the form of ferrous iron, and the most common site of absorption is proximal part of the small intestine, that is the duodenum. What are the factors that increases iron absorption? These are acidic pH, citric acid, vitamin C. Meat consumption can also increase iron absorption by increasing gastric acid secretion. Now the factors that are responsible to decrease iron absorption. So factors decreasing iron absorptions are alkaline pH. Acidic pH will increase iron absorption, whereas alkaline pH will decrease iron absorption. Iron absorption can be decreased by phytate, phosphate, tannate, milk, EDTA, calcium, and tetracycline. Iron is stored in the form of ferric iron, that is Fe3+, and they are stored in the reticuloendothelial cell, that is liver, spleen, and bone marrow. Hepcidin is a master regulator of iron absorption. Hepcidin is synthesized by liver, and the main function of hepcidin is it decreases iron absorption from the epithelial cells, and it will also cause decreased iron mobilization from the storage site. So this process is also important mechanism of development of anemia of chronic disease. Now we'll talk about iron absorption, its transportation and storage. So this is the lumen, this is the duodenal epithelial cell, and this side is the blood. So this duodenal epithelial cell, it has two surfaces, the apical surface or the brush border with villi, and it contains heme carrier protein, cytochrome B, and divalent metal transporter 1, that is DMT1, and the basolateral surface has ferroportin and hepastin. So this is the basolateral surface. So there are two types of dietary iron. There are two types of dietary iron, heme iron and non-heme iron. So heme iron is derived from hemoglobin and myoglobin of animal food source like meat, seafood and poultry. So it is the most easily absorbable form of iron, and it contributes to more than 10% of our total absorbed iron. So this is heme iron. And non-heme iron, which is inorganic iron, is derived from plant and iron fortified food, and it is less absorbed. So the absorption of most of the dietary iron occurs in proximal intestine, that is the duodenum, and it depends heavily on the physical state of iron atom. At physiological pH, that is in acidic environment, iron exists in oxidized form, that is ferric iron. To be absorbed, iron must be in ferrous form or bound by proteins such as heme. So the low pH of gastric acid will promote, or it will allow, a ferric reductase enzyme and duodenal cytochrome B present on the brush border of the enterocyte to convert the insoluble form, that is the ferric iron, is converted to ferrous iron, which is a soluble form or which is an absorbable form of iron. So gastric acid production plays a key role in plasma iron homeostasis. So when a proton pump inhibitor is given, such as omeprazole, iron absorption is greatly reduced. Once the ferric iron is reduced to ferrous iron, which is an absorbable form of iron, in the proximal duodenum, a protein on the apical membrane of enterocyte. So the protein on the apical membrane of the enterocyte, that is DMT1, divalent metal transporter 1. So it is a non-specific protein. So this protein will transport the ferrous iron across the apical membrane into the cell. So this protein will transfer the ferrous iron across the apical membrane into the cell, whereas the heme iron is transported across the apical membrane into the cell through heme carrier protein, that is HCP1. So once the iron comes inside the enterocyte, iron can be stored as ferritin or transported through the basolateral membrane into the circulation through ferroportin. So ferritin that is not bound to iron is called apoferritin, which has intrinsic catalyst activity that oxidizes iron into ferric iron to be bound and stored as ferritin. So what is ferritin? Ferritin is a hollow spherical protein consisting of 24 subunits that potentiates the storage and regulation of iron level within the body. So iron is stored in ferric form, that is Fe3+, on the inside of the ferritin sphere. So If iron is retained within the enterocyte and is not exported, it will be lost as mucosal shedding into the gut. So lifespan of the enterocyte is 2 to 4 days, and by mucosal shedding there will be loss of ferritin. It is also known as ferritin curtain. Which will finally get excreted in the feces. Now we will talk about iron transportation from duodenal epithelial cells into the blood. For transportation of iron in the blood, transferrin plays a major role. So transferrin is the major iron transporter protein. So iron binds to transferrin in the ferric form. Ferrous form of iron that was transported through the ferroportin must be oxidized to ferric iron. So this ferrous iron, it has to get converted to ferric iron so as to get transported by the transferrin. There are 2 copper containing protein that catalyzes this oxidation of ferrous iron. So ferrous iron gets oxidized to ferric iron by hepastin, which is present on the basolateral surface of enterocyte, while ceruloplasmin is the major copper transporter protein present in the blood. This hepastin is a primary protein that performs their function in a coupled manner with ferroportin, and it oxidizes ferrous form of iron into ferric form, whereas ceruloplasmin is involved in oxidizing ferrous form of iron into ferric form when the iron status is low. Now once oxidized, this ferric iron, it binds to transferrin and is transported to tissue cells that contains transferrin receptor. Here, binding of transferrin to transferrin receptor reduces affinity for iron. So iron is taken inside the cell by endocytosis and is stored in the monocyte macrophage system in the erythroid precursor cells of bone marrow. After the dissociation of iron from the transferrin is known as apotransferrin. This apotransferrin remains bound to its receptor because it has high affinity for its receptor at reduced pH. At neutral pH, apotransferrin dissociates from its receptor to enter into the circulation, and it will reload iron and it will repeat the cycle. In the erythroid precursor cell of bone marrow, this iron will be stored in the form of ferritin. So this will now form matured RBC, and the iron will be stored here in the form of ferritin, which will be seen in the serum as serum ferritin. So the serum ferritin is the marker of storage iron. Now the lifespan of RBC is 120 days. So RBC gets destroyed and iron is released, which enters into the blood vessel. Absorption of iron is controlled by hormone hepcidin. The liver has iron sensor. So when iron level gets high, this sensor signals for the release of hepcidin. This hepcidin is the master regulator of iron metabolism. Hepcidin effect on cellular level involves binding to ferroportin. This will result in internalization and degradation and effectively shunting the cellular iron into ferritin store and preventing its absorption into the blood. It also potentiates the excretion of iron through sloughing of enterocyte. So there will be mucosal shedding into the gut and finally into the feces out of the body. This was an overview of iron absorption, iron transportation and its storage. Now let's revise it once again. So there are 2 types of dietary iron, heme iron and the non-heme iron. Heme iron is absorbable form whereas the non-heme iron is non-absorbable. So heme iron is Fe2 plus and non-heme iron is Fe3 plus. So for absorption of iron into the cell, it requires the ferrous form and not the ferric form. Cytochrome B, it oxidizes ferric form of iron into ferrous form. Once the ferrous form is formed, it is transported across the cell by divalent metal transporter 1. So it is a non-specific metal transporter, as it can also transport iron, copper, lead and cadmium. So it is non-specific. Now this heme iron is also transported across the cell membrane by heme carrier protein, that is HCP1. Once inside the cell, they can either be stored as ferritin or they can be transported across the basolateral surface through ferroportin into the blood circulation. So for transportation of iron in the blood, we require transferrin protein, and transferrin protein carries ferric iron. So the ferrous iron that was transported through the ferroportin is now oxidized to ferric iron by hepastine or ceruloplasmin. Now this ferric iron will bind to transferrin protein and it will be taken to different tissues which have transferrin receptor, and the iron will be endocytosed into the cell, and it will be stored in the monocyte macrophage system in the erythroid precursor cells of the bone marrow. Now this will form matured RBC, and once the lifespan of RBC is completed in 120 days, RBC will be destroyed and iron is released, which will enter into the plasma cell or the blood vessels. Now this transferrin will also carry the iron to other reticuloendothelial cells like liver and the spleen. So here the iron will be stored in the form of ferritin. Now once the iron level is high, it will be sensed by the liver and it will produce hepcidin. This hepcidin will bind to ferroportin and will cause decreased absorption of iron and also the mucosal shedding, and ferritin will be lost in feces. So this was all about iron metabolism. Now we'll talk about lab findings. Now coming to the lab finding, the most sensitive and the earliest finding is decrease in serum ferritin level. So when there is decrease in iron storage, so there will be increase in transferrin level. When there is decrease in ferritin level, there will be increase in transferrin level, so that they can take more iron from the blood to the bone marrow. There will also be increase in total iron binding capacity. This total iron binding capacity is the total amount of transferrin that can combine with total amount of ferric iron. So the normal value of TIBC is 300 to 400 microgram per deciliter. In case of iron deficiency anemia, total iron binding capacity will be more than 400 microgram per deciliter. But in case of anemia of chronic disease, total iron binding capacity will be less than 300 microgram per deciliter. There will also be decrease in saturation. There will also be decrease in percentage saturation of transferrin because of increased transferrin and decreased iron level. In iron deficiency anemia, the percentage saturation transferrin level will be less than 15%. There will also be decrease in serum iron level, and so as to compensate the iron loss, there will be decrease in hepcidin level. When the hepcidin level decreases, there will be increased absorption of iron. So hepcidin level will enhance the iron absorption. Now what happens when iron deficiency anemia is associated with chronic inflammation like tuberculosis and cancer, etc. So when iron deficiency anemia is associated with chronic inflammation, there will be increase in serum ferritin. In iron deficiency alone, there will be decrease in serum ferritin. But when it is associated with chronic inflammation, there will be increase in serum ferritin level, because serum ferritin is also an acute phase reactant in chronic inflammation. Therefore, serum ferritin is of no use in diagnosis of iron deficiency anemia with chronic inflammation. Remember this point. So in this case, best investigation or gold standard investigation is bone marrow biopsy with pearl Prussian blue stain. And the second investigation is serum transferrin receptor level. So serum transferrin receptor will increase. So how will you differentiate iron deficiency anemia with chronic inflammation and anemia of chronic disease? In both these cases, there will be increase in serum ferritin level. So serum ferritin level is of no use in diagnosing iron deficiency anemia with chronic inflammation or anemia of chronic disease. So as to differentiate between these two conditions, the investigation of choice is serum transferrin receptor level. So there will be increase in serum transferrin receptor in case of iron deficiency anemia, in case of iron deficiency anemia with chronic inflammation, and serum transferrin receptor will be normal or decreased in case of anemia of chronic disease. This is all for today. I hope you understood the overall concept of iron deficiency anemia. So if you like the video, please do like and subscribe.